Globalization is confronting us with health and environmental challenges. Among the different approaches to these problems, rapid diagnosis stands out. COVID-19 spread is only one example of the urgent need for new rapid diagnostic techniques, which help fast, reliable and affordable diagnosis. And not only in healthcare, there are other threats like marine mycotoxins entering the food chain and the bloom of poisonous bacteria in freshwater reservoirs. And thus motivating is for me the reason of the work I present in this video. It is the result of a very close collaboration among material scientists, analytical chemists and biologists at the University of Oviedo. Rapid diagnostic testing is one way to refer to devices to detect viral proteins or antibodies generated by the patient's immune system. That is the case of COVID-19. The same type of rapid tests are demanded to detect other biomolecules like cancer biomarkers or toxins that can be in fresh water or foods or beverages. The World Health Organization encourages research to improve rapid diagnostic testing. Then, how can we make this technique better? Magnetism can be an answer. Our projects rely on these three pillars: the biological target that is determined by a microbiologist or a medical doctor, then the biofunctional nanostructures, which are the main focus of this talk, and the way to detect them, that is, the sensor which is sensitive to a property of the nanomaterial. All this aiming at rapid diagnostic testing for things like this. The idea of a rapid diagnostic test is not only that it is fast, which is clearly one of the points, but also that it is affordable, easy to use, maybe by untrained staff, easy to store and portable, then you can use it anywhere. Do you know any analytical device like this? I'm sure many of you know the classical home pregnancy test. This rapid test, as the COVID-19, is a lateral flow immunoassay. It is very easy to use. This end of the device is introduced in the biological fluid sample, and 10 minutes later we have one red line if the test is negative and 2 lines if we are pregnant. Now we need to put magnetic particles in these lines. Let me tell you what for. But first, how does a rapid diagnostic test work? The lateral flow immunoassay is a paper-supported system. It basically consists of a long strip of nitrocellulose membrane that acts as microfluidic platform, guiding the liquid sample by capillary action. We print a line of a biorecognition molecule across the strip. This is usually an antibody that selectively captures the molecule of interest. Then, a second line in which we use a non-selected antibody to capture anything biological that passes by. The target molecule is represented in this picture by a diamond and is contained in the body fluid, which is in the tube. We will pour in this tube the nanoparticles to tag the analyte. With this aim, the nanoparticles are biofunctionalized with another antibody also specific against the target diamonds. When the test strip N is introduced in the sample, the liquid flows along it by capillary action and the diamonds are captured at the test line, while the unbound particles proceed until they are stopped at the control line. The limit of detection in bioanalysis is a crucial parameter to assess the quality of the biodetection. It is defined as this ratio between sigma, which is the standard deviation of 10 measurements of the blank sample, and the slope of the calibration line. so the sensitivity. It describes the minimum amount of molecule that we can reliably detect. If in the commercial lateral flow immunoassay you see the lines in red color, this is because these tests use gold nanoparticles as labels, or other particles or enzymes which give color for detection. But we propose to substitute the plasmonic or the colored particles by magnetic ones. And why? Because magnetic nanoparticles can provide many advantages. We can use magnetic detection to increase the sensitivity. Look, while optical methods see the particles only at the paper surface, magnetic sensors can detect them in the whole volume across it. Also, quantifying them by optical methods is delicate and can be challenging, especially when there are optical interferences from the ambient light or the paper itself, which can be stained or maybe wet. Additionally, any detection method can benefit from magnetic immunoconcentration, so there is a lot of room for improvement. Before I go with those details, let me explain how much the magnetic particles weigh. And I'll talk about three main points, which are the capping, the aqueous suspension, and the magnetic properties that the particles must have. Look, the essential role of organic capping is to provide chemical endings to attach the biomolecules by covalent bond. This process is known as bioconjugation, and it is a key step in biodetection, of course. Additionally, particle coating can be a way to improve the surface layer of the particles. We know very well how much spin counting at the particle surface can spoil the magnetic properties. The magnetic dead layer disorder can be restored or partially restored by an adequate coating of the particle with an organic layer at high temperature. This is not a trivial job. I refer to the exciting work by Badger, Labatta and collaborators for this. In brief, we are seeing here the magnetite spinel structure that lacks some oxygen atoms at the surface. When we coat the particle with, for example, oleic acid, some of the acid's oxygens bond to the atom, resulting in an atomic configuration which is much closer to the bulk magnetite, and the outcome is a particle with improved crystallinity and magnetic properties. And the particle coating also contributes to the colloidal stability. The chemical groups endings provide electrostatic repulsion to counteract the van der Waals attraction. But for this, it is also crucial that the particles do not have a permanent magnetization. Then we go to the magnetic requirements. Then we need the particles to have no remnant magnetization for stability. We also need very susceptible particles to magnetize them easily at low cost. Finally, we need large saturation magnetization for good detection. All these requirements lead us to superpyramagnetic behavior, it has it all, and also superpyramagnetic sizes are small enough to interact very well with biomolecules. Immunomagnetic separation is a technique to isolate the cells or the biomolecule of interest out of the body fluid. Like this, we can separate the target molecule from a sample matrix, which can be complex and interfere with the rapid test. Besides, the same idea is tremendously helpful to increase the sensitivity by concentrating the analyte before running the test. And this is what we are using here. If we have a sample with a very low concentration, We start by adding the magnetic complexes to label the molecule of interest. We then incubate a few minutes and apply magnets. The magnetic particles drag the biomolecules towards the magnets. Then we can safely remove the supernatant, stir, and run the immunoassay. We are optimizing this technique for rapid and affordable biodetection of an infectious disease. Our hope is to get something useful for low resources locations or harsh environments, and which device can adapt to those requirements. Of course, the mobile telephone adapts very well to a lateral flow immunoassay technique. We can take a picture of the test paper and use, for example, image for evaluation. If we do not do any magnetic reconcentration, a limit of detection with this disease biomarker using the mobile telephone for calibration is 9 nanograms per milliliter. 9 nanograms of biomolecule per milliliter in the sample. If we use magnetic reconcentration, the limit of detection is reduced to only 0.6 nanograms per milliliter. So this is 9 and now we're here. This means that we are detecting only 60 picograms of this protein with a mobile telephone. We are very excited about this application. The mobile telephone device is obviously the most practical, but it will be insufficient in many cases. For example, if we need to determine the level of concentration of a substance. Quantification is not reliable in this case due to many optical interferences. The paper can be stained or the ambient light varies a lot. Given that our particles are magnetic, we use then a reader that is sensitive to their magnetic field. Among the options to detect superparamaneric particles, rapid diagnosis requires that it is a portable and affordable sensor. If possible, something robust and easy to use. It is why we have chosen magnetic inductive sensing, that is, for dielectromagnetic induction based on magnetically exciting the particles and detecting their magnetic induction. Now, to add complexity to the device, we have opted for planar coils that will act for both excitation and detection. For the same reason, the inductor is fed by a low amplitude current, and we continuously measure its impedance at radio frequency. We then slide the immunoassay strip over it in smooth contact, obtaining the impedance increase when the line passes on it. In a simple approach to it, we can calculate the initial magnetic susceptibility of a superparamaneric particle using Snell model like this. The DC magnetic susceptibility depending on the saturation magnetization and the anisotropy constant. Under 10, depending on the temperature and frequency like this. The frequency at which the susceptibility value decays, the characteristic resonance frequency, depends on the balance between the anisotropy energy and the thermal energy. On the one hand, the particle volume needs to be small for this to happen at radio frequency as we want to work. On the other hand, the susceptibility then grows proportional to the volume. Consequently, the ideal size is that at the threshold of superparamagnetism. This is with this model, the susceptibility in terms of the particle size. For magnetite at 20 MHz, the optimal size is about 12 nanometers. This is the experimental corroboration of this idea. We have studied different types of nanoparticles with sizes between 5 and 23 nanometers, obtained by different synthesis methods and with various coatings. And we see there is, as expected, a correlation between the sensitivity and the initial susceptibility, which is here represented in the x-axis. Accordingly, the resolution is reduced and the higher susceptibility particles allow detecting smaller amounts. Once the signal per particle is optimized, we can use clustering or encapsulation in polymeric vesicles to increase the signal per molecule. So here there is a step of bioconjugation, and it involves the number of particles that we are able to selectively attach to a molecule. This is a measurement with the inductive sensor of a lateral flow immunoassay made with 12 nanometers magnetite particles. I'm not giving details on the biomolecule here. 12 nanometers is the ideal particle size. And here is the same experiment but using clusters made of those same particles. Of course, the signal is very much enhanced. Biodetection benefits from clustering because the number of particles per molecule is increased, and so does the detected signal. The limit for the cluster size is the pore of the lateral flow membrane. And for the same reason, clustering is also beneficial for immunomagnetic separation. I would like to show you some results on two biodetection problems. The first of them is related to health. Imagine that, for any reason, your physician suspects that you might be suffering colorectal cancer. Then they will prescribe you a fecal occult blood test. This is a very sensitive technique, but very unspecific. It can be positive for many benign reasons. And if it is, the physician will strongly advise that you undergo a colonoscopy. But colonoscopies are invasive. Many people do not want to do it, and many can just not afford it. So what if we had some tool in the middle to avoid a lot of maybe unnecessary colonoscopies? The microbiologists working this project have advised to look for exosomes. Exosomes are very much in fashion these days because they are potential biomarkers for many diseases and specifically for many cancers. They are vesicles that the cells release to the bloodstream and contain information which is characteristic of their cell of origin. They are a little bit like our garbage bags that can tell a lot about us. Inside the exosomes, there is RNA and DNA. Please pay attention to the exosome surface. These in green are proteins. CD81, 9 and 63 appear in all exosomes, no matter their origin. CD147 can be, according to recent studies, specific of colorectal cancer. We will then use CD9 to capture the exosomes in the lateral flow strip, and CD63 determine the number of exosomes in the sample. And then we will label with 147, trying to identify colorectal tumors. These are some of the lateral flow strips that we obtained for calibration. And these are the calibration curves. In the x-axis, the number of exosomes 10 to 7 per microliter. The curves in black are the measurements with the Faraday inductive sensor. In color, the lines obtained with a commercial reflectance reader. This is a desktop optical device. First thing to look at is the linear range. For the magnetic sensor, this is all the measured range, while in the optical sensor, the range is shorter in both graphs. The reason is the saturation of the immunoassay In the test line, there is, of course, a finite number of antibodies. When all the sites are occupied, the exosomes that get there must cross without attaching. Of course, the surface layer of the paper will saturate before the whole volume, and this is why the optical signal saturates before. The inductive sensor also has a better correlation factor and a smaller limit of detection in both graphs. The magnetic detection labels used in these experiments were multi-core particles consisting of magnetite superpramagnetic nanoparticles coated by a double layer of oleic acid. These strips were obtained in a purified sample. Purification from human plasma is done by ultracentrifugation, filtration, and other washing procedures. The lateral flow immunoassay works for both gold nanoparticles and oleic acid magnetite particles. On the right, we performed the assays introducing the paper directly in human plasma from the hospital patients without purification. As you can see, it didn't work well with gold nanoparticles. We see no test line at all. But it did work fine with oleic acid coated particles. No sample pretreatment is needed then, and this is excellent news to achieve a rapid diagnostic test. The second type of application example I want to show you today is the problem of food safety. Foods and beverages that undergo fermentation, either in their production, like wine, beer or chocolate, or at the beginning of their decomposition, may contain a molecule called histamine. Large amounts of histamine may be lethal, but for some people, even small amounts can produce headache and other intoxication symptoms. I am one of those persons, and I love wine. I engaged enthusiastically in a project to detect histamine in red wine. Here you can see the magnetic lateral flow immunoassay. The type of immunoassay is different in this case, and the intensity of the test line decreases with the amount of histamine. This is a competitive immunoassay in which the histamine competes for deposition with histamine immobilized in the test line. In the right image I want to compare lateral flow strip for red wine made directly with gold nanoparticles here on the right, and with oleic acid coated magnetite nanoparticles here on the left. You can see how clean the strip of the left is. The oleic acid helps the matrix of the sample flow, so the paper is much cleaner here. It is impossible to quantify with gold nanoparticles, which are the same color as red wine. So, with this bioconjugation, we prefer magnetite. The magnetic lateral flow immunoassays were calibrated by optical and magnetic readers. Again, the inductive sensor, here in black color, produces better parameters than the reflectance reader. It also produces better linearity and limit of detection. Here is the validation of our results against ultra-high performance liquid chromatography. This was made on wines in different production stages. Look how good the easy and fast magnetic lateral flow immunoassay is if we compare it with the sensitive but sophisticated and expensive technique. To wrap up, Can we use magnetism to develop a breakthrough type of rapid diagnostic test technology? Absolutely, I am convinced that the answer is yes. In this way, and probably with other approaches, it pushes us to make a lot of magnetic research, to reduce the particle magnetic anisotropy, to further increase the radio frequency susceptibility, to study the interparticle interactions within vesicles, the particle interaction with the biomolecule, and the particle interactions with the sensing device, to develop a practical, sensitive technology to face crucial challenges in healthcare, environmental, and food control. I would like to give credit to our collaborators in these projects, and of course to the funding agencies. And thank you for watching this video.